So, good morning, students.
Good morning, sir.
So, today we are going to start the new genus, or you can say the second last genus of our syllabus. So, Ectocarpus systematic position as per Fritsch. You knew that out of those 11 class, Fritsch placed this uh, algae at this seaweed in Phyophyceae under order Ectocarpus, family Ectocarpaceae, and genus Ectocarpus. Whereas, like other Xanthophycean and Phyophycean algae, because of their heterocontype flagella, Lee placed Ectocarpus under phylum Heterocontophyta, and in that phylum he placed this under Phyophyceae. Okay, so now you can understand why Xanthophycean and Phyophycean algae are close to each other. And the rest of the things were same: order Ectocarpus, family Ectocarpaceae, genus Ectocarpus. So just have a quick revision about Phyophyceae before starting the Ectocarpus. The most important thing that they are almost marine, means they are very rare things which you will find in fresh water. Mainly grow in intertidal belt and upper littoral zone. अदरवाइज आप डीप वाटर में जाएंगे देन यू विल फाइंड द रेड एल्गी, और जो एक्सपोज एरिया है जो सी का, देन यू विल फाइंड मेनली ग्रीन एल्गी. अब अलोंग विद क्लोरोफिल आप याद रखिए कि नो मैटर whether it's algae, bryo, tido, gymno, angio, monocote, dicote, whatever it is, the ultimate जो आपका photosynthetic pigment है, that is chlorophyll A. And there are some accessory pigment like chlorophyll C, carotene, and fucoxanthin. And because of this fucoxanthin, this the overall color is coming out as brown color, different shades of brown color. That's why all these algae are known as brown algae. Reserve food materials are laminarin, mannitol, and fats. तो अभी तक आपने पायोनोड्स देखे होंगे inside the chloroplast, but here there are certain things where you can see that pionoids are attached with the chloroplast with a stalk. Cell wall जो बनी होती है that is mainly cellulose, fucodine and alginic acid. Alginic acid क्या होता है? It is a polymer of mannuronic acid and gluuronic acid. इसके अलावा हमारे डी मैनिटोल इज द एक्यूमुलेशन प्रोडक्ट ऑफ फोटोसिंथेसिस. कई बार आपके मन में क्वेश्चन आज आया होगा या नहीं आया, तो अब जरूर लाइएगा कि व्हेन वी आर हैविंग laminarian, mannitol, alginate, means why there are different kind of sugars or carbohydrates are present. So whether it's laminarian or alginate, they are complex carbohydrate. Alginic acid, you can say they are the part of their cell wall, और या आप ये बोल सकते हैं it will give the structure to the cell, whereas mannitol will help in the transportation of the sugar. तो जो डिजोल्व होकर सेल्स के अंदर ट्रांसपोर्ट करती है सुगर, वो मैनिटोल है आपकी, और जो स्ट्रक्चरली एक जगह कन्फाइंड रहती है इन द सेल वॉल, दैट इज एल्जिनिक एसिड. इसके अलावा लेमिनेरियन भी आपको मिलता है, समटाइम्स फैट्स आल्सो, बट mainly it's reserved food material. In vacuole you will find laminarin, in cell wall you will find your alginic acid, and in between the cytoplasm wherever it is required, mainly inside the mitochondria, it is D-mannitol. So as you knew that Lee का पूरा classification ही based है how many endoplasmic reticulum and chloroplast envelopes are there, along with the number of thylakoids. तो आपको ये तो पता लगेगा there are two membrane of endoplasmic reticulum which are in continuous with the chloroplast, as well as you will see 3 thylakoid per band. जो भी आप chloroplast के band देखेंगे, you will see there are 3 thylakoids stacked one over other, and that's why all those algae which are having 2 endoplasmic membrane along with 3 thylakoid per band will come under Phyophyceae. Phlorotannins, ये आपका Phyophycean tannins को बोलते हैं. It get formed by Golgi body in and stored in phycode in the cytoplasm of many brown algae. So this tannin will give them a bitter taste to all the brown algae. That's why they can protect themselves from the herbivores. Okay. इसके अलावा अल्ट्रावायलेट रेडिएशन पे भी बचाते हैं, and as well as they also serve as the component of cell walls. Already discussed from microscopic to macroscopic, but in general they are macroscopic. Flagella only present in zoospore. जैसे हमने वाउचीरिया में मेन थैलस में हमें फ्लैजेला नहीं दिखा था, in the same way the main thallus don't have flagella, whereas flagella or motile stage restricted only to zoospores and gametes. And if there are flagella, there are two kinds of flagella: long anterior tinsel flagellum and tripartite hair, and a shorter posteriorly direct whipless flagellum. The most complex flagella जो आप देख सकते हो, you will find in Phaeophysi. Whereas the fucales are an exception in this, with the posterior, जो हम कल बताएंगे इसके बारे में, with the posterior flagellum of the spermatozoids being longer than the anterior. Otherwise, whether it's Ectocarpus, Laminarials, and all other order, it is vice versa. Eye spots is also present around 40 to 80 lipid globule के साथ. Sexual reproduction is also very complex, starting from isogamous to oogamous. The cell structure is in many ways similar to that Chrysophysi, Perminophysi, Basidiophysi, and Xanthophysi, with a main difference of large amount of extracellular polysaccharides surrounding the protoplast. आप ये जितनी भी क्लास पढ़ रहे हैं, मैं ली की बात कर रहा हूँ, all or more or less they are same, जितना भी आप पढ़ रहे हैं, but their main difference is you will find mainly in their cell wall. जैसे ब्राउन एल्गी में हमारे आपके कोर्स में ब्राउन एल्गी ही है, तो उस ब्राउन एल्गी में you will find mainly the alginic acid. In other groups or the other class of this phylum, you will not find alginic acid. So these were the basic characteristic features of brown algae. Is it clear to everyone?
Yes, sir.
Yes, sir.
This is the diagrammatic representation of the brown algal cell. ऐसा आपको cell नहीं दिखाई देगी कभी भी, but it is just showing के what are the different components you will find in a brown algae. इतना prominently आपको chloroplast नहीं दिखाई देगा, इतना prominent आपको pynoids नहीं दिखाई देंगे. It's a diagrammatic representation. That if you will enlarge the pynoids, you will see that it remain attached with the chloroplast with the stalk. और इसके बाहर आप अगर chloroplast को enlarge करके देखेंगे, then you will see there are two membrane along with the chloroplast of endoplasmic reticulum. और ये साथ में nucleus के साथ continuity है. आप ये ध्यान दीजिए, nucleus then chloroplast, and in its continuity there are two membrane of endoplasmic reticulum. हो सकता है बहुत सारे क्लोरोप्लास्ट हो, chromatophores हो, और they are different structures of chromatophore, but they are in continuity with the nucleus, so that whatever mRNA is producing, it will directly come in the contact of chloroplast. उसको ट्रेवल में टाइम ना लगे. अपने नेचर बहुत ब्यूटीफुल है कि सोचो कि कितना स्पेसिफिक तरीके से बनाया, because in between, if chloroplast and nucleus are far away from each other, and if there is no connective link of the endoplasmic reticulum, तो जो ट्रांसक्रिप्शन होने के बाद प्रोटीन बनाना है, suppose it's an emergency protein is required, so in transportation of that mRNA from the nucleus to the ribosomal site, they may get some kind of damage. To protect them from that kind of damage, nature make this system that in brown algae and in many of the eukaryotic cells, mitochondria, endoplasmic reticulum, chloroplast, they are always near to nucleus. जो गोल्जी बॉडी होता है आपका, it is slightly away from this, so that it can excrete out whatever the remaining leftover after the their function of that protein. Ectocarpus के main features क्या होते हैं? Thallus consisting of filament or filament compacted together. और इसी वजह से it give us a pseudo parenchymatous appearance. Let's start discussing the ectocarpus. If we see that this is one of the most common species is Ectocarpus. What's the name of the species? You can see on your screen.
Ectocarpus siliculus.
Siliculosis. It's a cosmopolitan marine algae. Okay. Commonly found as free floating filament in the intertidal zone to sublittoral zone on the shore. It can easily grow as epiphyte or endophyte. And if you want to know more about this algae, starting from its life cycle to proteomics, genomics, everything, you can get all the information from Dr. Alokar. And whatever text is there, it is mainly from the chapter written by Dr. Alokar. When he tried to culture it, if it is a gametophytic structure, it was normally a floating, and when he was able to get it the sporophytic structure of ectocarpus, it was normally attached. But in general, आप वहाँ पे सर्च करेंगे नेचर में, then it is very difficult for you to differentiate between a gametophytic and sporophytic thallus. And if it is so, then both kind of thallus will known as what kind of thallus? Isomorphic or heteromorphic?
Isomorphic.
Yes,
Isomorphic.
isomorphic. When we cannot differentiate between gametophytic and sporophytic condition. That's why their life cycle is of isomorphic type. Because most of the hormones we have studied in algae and any other plant, this is one of those model organism where we have studied the different pheromones and their physiological process. So you can see it's a very old material of Ectocarpus. And again, I'm repeating this again and again. Whenever you are taking any material, try to take as less material as possible. जितना आप कम लेंगे उतना अच्छा होगा. Just have a look. कि कितना सा मटेरियल मैंने लिया है. Is it visible to everyone?
Yes, sir.
Then why we are using safranin? Because it don't have starch as storage material. That's why there is no need to use iodine. And it will take a rapid stain of safranin. That's why immediately you have to take it out after staining it with safranin. Then transfer it in glycerin, which is a mounting material, and very gently. बहुत ध्यान से आपको ये करना होगा so that each and every filament of this algae will get separate out from each other.
Yes, sir.
And whenever you will place the cover slip over the material, make sure that you will gently place it in 45-degree angle and gently remove the needle or whatever things you are having, so that there is no air bubble will be inside your cover slip. Now we will observe it under the microscope. The thallus is heterotrichous. What is the meaning of heterotrichous?
सर इरेक्ट सिस्टम और सर जिसमें वो होते हैं सर राइजोडियल नीचे और पूरा मतलब एक इरेक्ट सिस्टम होता है हाईली ब्रांच्ड.
Algae is one of the most.
Thallus is
Bolo, thallus is
The thallus is very much evolved and differentiated into prostate and erect systems.
बिल्कुल ये this is the main difference. When the thallus of any organism is differentiated in two systems. One is prostate and another is erect. The higher plant are having root system and the shoot system or the stem system. Similarly, if algae are have been differentiated the entire thallus in two parts, the rhizoidal system and the erect projectile system, that condition is known as heterotrichous condition. जैसे क्लेमाइडोमोनास आप देखेंगे, वोल्वोक्स देखेंगे, you will not find a rhizoidal kind of structure over there. So those algae are not heterotrichous, whereas Ectocarpus, Polysyphonia, and many others, they are having the rhizoidal outgrowth. So that's why they are known as heterotrichous algae. If we talk about its cell, it's a filamentous algae, so cells are arranged one after the other in a chain. And inside the cell, you will see a nucleus with many chromatophores, discoid और band shaped. यहाँ पे हमें इसका जब स्ट्रक्चर आपको दिख रहा है, then you can have a look. You will see there is very irregular shape of chromatophore or chloroplast आप बोल सकते हैं. You will see there is no definite structure. This is different in this cell, different altogether different in this cell, and different in this cell. Can you see this?
Yes, sir.
Okay, so this is the wrong diagram. आपको ये इस तरह का डायग्राम नहीं बनाना चाहिए. In many books you will see this kind of diagram. Actual in Ectocarpus, there is no definite structure of the chloroplast or chromatophores, and they are always surrounding the nucleus. जो बीच का जो nucleus part है यहाँ पर है, this is nucleus over here. Okay, so they are always near to this, and actually they will attach themselves with the nucleus with the help of endoplasmic reticulum. In continuity में होते हैं ये. But जब हम इसको ऐसे नेचर में देखते हैं तो sometimes we are not able to see that connection between nucleus and endoplasmic reticulum. But yes, if you will go in the detail or ultrastructure, you will find the direct connection of nucleus with the chromatophores or chloroplast with the help of endoplasmic reticulum. And in between them you will see projecting pyronoids. अभी तो आपको नहीं दिख रहे होंगे ये जो pyronoids, you can see the pyronoids, but you cannot observe them whether they are attached with the help of struct or not. But if you will go for the ultrastructure, then you will be able to see it. Green color of chlorophyll A and C is completely masked by high amount of xanthophyll. Xanthophyll ज़्यादा होता है, chlorophyll कम होता है, and it depends कि if in X algae chlorophyll is comparatively more, so it will give more greener color. If it is xanthophyll more in that algae, then it will give more brown color. So different shades of brown इसी वजह से होते हैं because they are having different amount or different concentration of chlorophyll A and C along with xanthophyll. Lysosomes like vacuoles present in the cytoplasm. Growth is trichothalic. ये important terms है. What does it mean? That with intercalary cell division confined to certain areas of the filament. Okay, cell division is restricted to the base of one or several filaments. I will show you in the end के क्या है, but अभी मैं एक बार दिखा देता हूँ आपको. You will see that there are certain points from where the branches are arising. Although it's highly branched, but you will see there ऐसा नहीं है कि कहीं से भी ब्रांच हो रही है. इस एरिया से ब्रांच निकल रही है, कुछ इस एरिया से ब्रांच निकल रही है. So there are certain points. It's not dichotomously branched. Joining dichotomous हो, it may have an irregular type of branching system, and that's why it is this kind of growth is known as trichothalic kind of growth. Scientists divide up the family into different genera. बहुत सारे साइंटिस्ट ने बोला है कि yes, there are different genera, but at present, most of the phycologists believe that Ectocarpus is the lone genus of family Ectocarpaceae. अब हम मॉर्फोलॉजी देखते हैं कैसे में दिखाई देता है। दिस इज द सेम थिंग्स, जस्ट हैव ए लुक, कितना फिलामेंटस है, हाईली ब्रांच। This is the way you have to observe it. Sometimes students get confused in ectocarpus and polysiphonia. Just try to enlarge this any part of this screen. एक बार enlarge कीजिए. You will see that each and every filament is a tube-like structure, single tube-like structure. Just please note down this point. आपको ये note करना है just for your understanding. You will not write this point in your record file or in exam. And if you will see that it is again differentiated into many tube, like एक के बाद एक tube से बनी हुई है, siphon like structure से बनी हुई है, then it is polysiphonia. Are you getting my point? If it is made up of only single tube like this, it is ectocarpus. If it is made up of many tubes, it is polysiphonia. Can you see this? Here you will see there are more than one tube like structure, polytube like structure.
Yes.
Can you see it?
Yes, sir.
Yes, sir.
That's the main difference. ये आपको ध्यान रखना है कि अगर हमारा एक्टोकार्पस है तो आप इसे कितना भी इनलार्ज कर लीजिए, you can not differentiate कि इसके अंदर एक ट्यूब हो रहा है. They are made up of single row of chain cells, single, क्या बोल रहा हूँ मैं, sorry. You will see it's a chain of single row of cells. एक के बाद एक सेल जुड़ी हुई है चेन के रूप में. लेकिन अगर आप सेम टाइम पे अगर आप पोलीसाइफोनिया को ऑब्जर्व करेंगे, you will see there are tube like structure in between. दिख रही है ना, एक से ज्यादा आपको स्ट्रक्चर दिखाई देंगे. यहाँ पे देखिए. Okay, that's why it is known as polysiphonia. But if you are getting confused in between whether it's ectocarpus or it is polysiphonia, so make it a thumb rule. Try to focus on a single filament. If it is made up of only one thick cell, it is ectocarpus. If it is there are many polytube-like structure, it is polysiphonia. यही यहाँ पे भी क्लियर है काफी, मैं वैसे ही गया उधर. Here you can see clearly. Okay. आपको कुछ और स्ट्रक्चर भी दिखाई दे रही होंगी लाइक दिस, बट वी विल डिस्कस इट लेटर ऑन. आज ही करेंगे बट थोड़ी देर बाद. Okay, you cannot see any kind of tube-like structure inside a single filament. Now let's start discussing the life cycle. So it is a alternating multicellular heteromorphic gametophyte and sporophyte generation. I am saying that there are two contradictory statements. In initial of this lecture, I said that it's a isomorphic type of life cycle. Here I am writing it's heteromorphic gametophyte and sporophyte. Why it is so? अगर मैं एक्सप्लेन ना करूं तो आप मुझसे प्लीज पूछिएगा ये बात कि सर आपने दो अलग अलग चीजें एक ही जीनस में आपने हमें क्यों बताई. आई विल एक्सप्लेन इट. अभी करूंगा तो थोड़ा कंफ्यूजन हो जाएगा. आफ्टर 10 मिनट यू विल अंडरस्टैंड इट. The haploid and diploid phase are both filamentous, but the diploid filament have longer cells than the haploid filament. अब आप देखिए यहीं पे आपको एक पॉइंट मिल गया है. The diploid filaments have longer cells than the haploid filament. It means they are morphologically different. But to observe that morphology, you have to use माइक्रोस्कोप. आर यू गेटिंग माई पॉइंट? कि आपको सेल्स की साइज देखना है तो आपको अंडर द माइक्रोस्कोप देखना पड़ेगा, एंड यू हैव टू ऑब्जर्व इट वेरी केयरफुली. यस और नो?
Yes, sir.
क्योंकि तभी तो जब आप दोनों सेल्स को देखोगे देन ओनली यू कैन कंपेयर के एक सेल्स बड़ी है और एक छोटी है. So if you think that this is a feature, we can say it's heteromorphic. You can consider it's heteromorphic. If you will say कि नहीं देखने में तो एक जैसे लग रहे हैं दोनों sporophyte and gametophyte, then you can say it's isomorphic. If you are going in that much detail where you can compare the cell size, then you can say it's heteromorphic type of life cycle. क्योंकि दोनों बिल्कुल अलग अलग हमें दिख रहे हैं. Gametophyte की cells अलग है, sporophytes की cells अलग है. That's why they are heteromorphic. लेकिन अगर किसी ने superficially normal microscope से देखा है तो he or she will not be able to differentiate it neither in nature nor in culture. That's why they can say they are isomorphic type of life cycle.
Yes, sir.
Yes, sir.
Okay. The sexual life cycle in the sporophytic generation start with the formation of a diploid zygote. Zygote से हम मानते हैं स्टार्ट होती है और उसके बाद हम बात करते हैं कि that zygote will germinate and will give rise to sporophyte, and that sporophyte will give rise to gametophyte. How it will happen, that we will discuss later on. If we are observing the sporophyte, then only we can see the heterotrichous condition. Otherwise हमें तो normal floating condition में ही gametophyte मिलेगा. And two specialized reproductive structure, plurilocular and unilocular sporangia. This is plurilocular sporangia, this is unilocular sporangia. Unilocular, 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 plurilocular sporangia. This is the still photograph of the permanent slide, which is showing the plurilocular sporangia. This is the enlarged view of plurilocular sporangia. Can observe it under the microscope. Permanent slides का ही है ये. As you can see, this is full of unilocular sporangia. यूनिलोक्युलर क्यों? क्योंकि इसके अंदर एक ही सेल्स की बनी हुई होती है, एक मेन सेल होता है इसके अंदर. एंड इफ यू विल see, when you will see the plurilocular, it is made up of different chamber inside the same structure. These are the plurilocular sporangia. You can see it is made up of different small structures. Can you see it? आपको दिखाई दे रहा है ये?
Yes, sir.
If it is made up of many structures, small boxes like structure, it is plurilocular sporangia. If it is a single and large one, that is unilocular sporangia. And with the presence of plurilocular sporangia, you can also easily identify the ectocarpus. याद रखना कि when you will discuss the polycyphonia, you will ask us what is the major difference between the plurilocular sporangia and the reproductive structure of polycyphonia. I am willingly not taking the name of that structure so that you will not get confused. This is the life cycle, and with the help of this photograph, I will try to explain the entire life cycle. Plurilocular sporangia lead mitospores, okay, or mitozospore. What is the meaning of mitozospore? They are forming after the mitosis only. If it is a product of meiosis, they are known as meiospore or meiozospore. It means this is our starting point. We are starting it from here. What is happening in entire life cycle? This plurilocular sporangia will undergo mitosis and will produce mitospore or mitozospore, and they will keep on germinating and they will produce similar sporophyte. एक के बाद एक जनरेशन asexual reproduction करते चले जाएंगे. Whereas unilocular sporangia single chamber, ये जो that contain meiotically produced meiospore or meiozospores and give rise to multicellular gametophyte. And these spores will come out and germinate and will give rise to gametophyte. अब आप देखेंगे this structure and this structure is same. Morphologically, they are completely same. The only difference, one of these is having slightly longer cell. As well as if you are culturing sporophytic ectocarpus and gametophytic ectocarpus, sporophytic ectocarpus you will see in the bottom of your culture, whereas gametophytic structure you will find in floating condition. One more thing, unilocular sporangia is also present only in sporophytic condition. So there are key difference between the gametophyte and sporophyte of ectocarpus, but आप मॉर्फोलॉजी के बेस पे आप उसको डिफरेंशिएट नहीं कर सकते नेक्ड आई से या बाय विद द हेल्प ऑफ सिंपल माइक्रोस्कोप. इट्स वेरी डिफिकल्ट. आपने सपोज कीजिए ये वाला स्पोरोफाइट लिया और इस पोर्शन को ऑब्जर्व कर रहे हैं वेयर देयर इज नो यूनिलोक्युलर स्पोरेंजिया एंड यू आर ऑब्जर्विंग दिस गैमेटोफाइट, बोथ आर सेम until unless you are not focusing on the cell's length, कि सेल की लेंथ कितनी है दोनों में, you cannot differentiate. That's why some scientists claiming that this kind of life cycle is a isomorphic type of life cycle, and some is believing that's a heteromorphic type of life cycle. When we will observe the ectocarpus, there are also difference between the species of the same ectocarpus. कि हो सकता है कि कुछ में cells का difference बहुत ज़्यादा हो. Then we can say that this species is showing the heteromorphic type life cycle, and in other species where the cell size is not that prominently different from each other, where there at that time we will say it's showing the isomorphic type of life cycle.
Yes, sir.
Is it clear?
Yes, sir.
चलिए थोड़ा सा मैं कहीं कहीं पे बीच में एडिशनल इंफॉर्मेशन दे रहा हूँ इसमें, but it's not for your theory purpose. But if you are interested, the text which I have mentioned in italics, it's an additional information. When unilocular sporangia will produce the myozospore, they are generally 32. और क्योंकि वे म्यो है तो automatically they are haploid in condition. It may be A4, it may be 8, it may be 16, it may be 32. And then they will come out, okay, and you again, whether it's the myozospore or this mitozospore, morphologically they are similar. The haploid spores, they are relatively large compared to diploid spores. This zoospore initially swim in a straight pattern. It will swim, and it will, when it will get its appropriate substratum, mainly hydrophobic surface or a surface where you will see microbial growth, and on that surface it will germinate. This zoospore will germinate and will give rise to this gametophytic condition. यहाँ पर लिखा है morphologically it is hard to distinguish between a male and a female gametophyte as well. Male gametophyte and female gametophyte है, we cannot even differentiate between them. और मेल और फीमेल gametophyte के बेस पे भी हम उसको heteromorphic बोल सकते हैं. Only upright filament that I have already explained. स्पोरोफाइट में तो आपको प्रोस्टेट सिस्टम मिलेगा, लेकिन गैमेटोफाइट में आपको प्रोस्टेट सिस्टम नहीं मिलेगा. ओनली फ्लोटिंग कंडीशन यू विल सी. इफ यू आर कल्चरिंग इट एंड आपको लगता है कि ये फ्लोटिंग कंडीशन में है, देन यू कैन से यस इट्स मोस्ट प्रोबेबली द गैमेटोफिटिक स्ट्रक्चर. इफ इट इज अटैच्ड देन यू कैन से यस इट इज स्पोरोफिटिक. और चाहे ये गैमेटोफाइट है, सिमिलर काइंड ऑफ प्लूरिलोकुलर गैमेटेंजिया. अब हम यहाँ पे स्पोरेंजिया नहीं बोल रहे हैं, गैमेटेंजिया बोल रहे हैं, बिकॉज इट्स गैमेटोफाइट. Plurilocular gametangia is also produced, which is almost same like the plurilocular sporangia. काफी बुक में इसको sporangia ही लिख देते हैं. यहाँ पर भी sporangia लिखा है, यहाँ पे भी sporangia लिखा है, but I willingly change its gametangia. There are around 660 cubical cells. That's why it is known as plurilocular. Again, there is one more additional information. Some of the unfused gametes have the ability to germinate parthenogenetically. अब ये जो gametes हैं हमारे, this gametophyte will produce male gamete and female gamete. They will get fused, but some of these gametes may develop in the same kind of gametophyte with the process which is known as parthenogenesis. पार्थोजेनेसिस मीन्स कि हमारा जो गैमेटोफाइट है इट्स नॉट ए प्रोडक्ट ऑफ द म्यूजिस्पोर, इट्स ए प्रोडक्ट ऑफ फीमेल गैमीट। तो अगर कोई पूछता है व्हाट द मीनिंग ऑफ पार्थोजेनेसिस इन एक्टोकार्पस, you will say when gametophytic structure is developing from the female gamete instead of mitozois or myozospore, then this is known as parthenogenesis. The female gamete settle down about 5 minutes after liberation and it will liberate a secrete a sex hormone, उसको बोलते हैं ectocarpin, and this ectocarpin will attract the male gamete, और मेल गैमेट विल मूव वेरी रैपिडली 269 माइक्रोमीटर पर सेकेंड। आप सोचिए प्लांट के केस में रैपिड का मतलब क्या होता है, 269 माइक्रोमीटर पर सेकेंड, कितना हुआ ये 269 माइक्रोमीटर पर सेकेंड का मतलब हो गया 1 मिलीमीटर डिस्टेंस को कवर करने में तो पूरे 4 सेकेंड लग रहे हैं। तब भी प्लांट के केस में हम वेरी रैपिड बोलते हैं क्योंकि प्लांट्स आर नॉर्मली नॉन मोटाइल। And many male gametes cluster around a female gamete and attach to it by their longer anterior flagella, and they will make a special structure which is known as clump formation. Please note down this. Almost every year, directly or indirectly, examiner is asking what do you write a short note on clump formation. So what you will say in ectocarpus, when female gametes get released from the female gametophyte, it will get settled down and it will release ectocarpin hormone which will attract the male gamete, and many male gamete will get attracted toward this female gamete and they will get attached by their long flagella and in a group, and then they will form the structure which will form will known as clump formation. इसको हम बोलते हैं clump formation. The motile male gamete which can remain motile for up to 8 hours swim in circular path because they are getting attracted because of this ectocarpin. So to kitni small structure, still they are having that sense that from which side this ectocarpin is being released. So to water mein, it's not in environment, it's inside water, but still that small amount of ectocarpin can attract the male gamete. And the more they will come close to this female gamete, their speed will keep on slowing down. पहले बहुत फास्ट मूव करेंगे and they will try to search out कि कहाँ पे हमें ectocarpin मिल रहा है, and then the moment they will get any sense, okay, this is the direction from where we are getting this ectocarpin, so they will start rotating themselves around this female gamete. और जब एक साथ सारे आके से जुड़ जाएंगे then they will form clump formation. One of these male gametes will get fused with this female gamete. Before they get fused, they will get fused from the same area where the flagella was present, and after fusion they will form a diploid zygote. और zygote बनने के बाद before they germinate, they will release their completely their flagella. Okay, one more thing. The process of fusion takes place about 20 seconds, and after fusion the zygote loses its attraction for the male gamete. Once one of these get fused, okay, then it will not stop production of ectocarpin. और ectocarpin को देगा production बंद कर देगा, then the rest of the male gamete will detach themselves from the male gamete and then it will germinate within 2 to 3 days. तो हमने जहां से साइकिल स्टार्ट की थी, from this zygote we started from those sporophytes. These sporophytes are having this plurilocular sporangia which keep on giving rise to the mitospore or mitozospore, and वे एक के बाद एक multiple अपने sporophyte बनाते जाएंगे, and those sporophytes may also develop unilocular sporangium. Those unilocular sporangium after the meiosis may produce up to 32 meiozospore, which after germination can give rise to the gametophyte. Male and female gametophyte are similar to each other. And fusion कैसे होगा? Because female gametes will get settled down, start releasing the ectocarpin, start attracting the female gametes. Male gametes start rotating themselves around the female gamete, and one of them will be successful to get fertilized itself with the female gamete, and that will form zygote. But उस केस में क्या होता है, कई बार में जो gametes होते हैं, female gamete या male gamete, they can also give directly germinate into gametophyte, and this process is known as parthenogenesis. ये आपके माइटोजूस्पोर हैं या ये आपके म्योजूस्पोर हैं, और ये जो गैमीट्स हैं, all these are example of asexual spore. क्योंकि ये जो गैमीट्स का जो फ्यूजन हो रहा है, this is the only sexual part. In what other manner they are developing, whether in sporophyte or gametophyte, they are part of asexual reproduction. लेकिन asexual reproduction and sexual reproduction is entangled with each other. इतने बुरी तरह से मिक्स एंड मैच है एक्टोकार्पस में कि हम उसे asexual अलग से और sexual अलग से नहीं पढ़ सकते. हमें पूरी लाइफ साइकिल के साथ ही समझना होगा what are those things which are germinating asexually and what are those things which are germinating after a sexual reproduction. तो ये हमने यही इसमें बताया आपको कि कौन कौन से अलग मोड है asexual के. We gamete that fail to find a suitable partner, mitotic event in plurilocular sporangia form mitospore, or sometimes myospore released from the unilocular sporangia can change their fate to develop into sporophyte. This particular phenomena where a myospore develop into sporophyte is called heteroblasting. तो ये 3-4 अलग-अलग तरीके हैं जो आपके you can consider them that they are the method of asexual reproduction. एक बार और रिपीट कर दो पूरी लाइफ साइकिल.
Yes, sir.
हाँ, थोड़ा ध्यान से सुन. What we are saying, we are starting from this zygote. हम कहीं ना कहीं से तो स्टार्ट, सर्कल को कहीं ना कहीं से स्टार्ट मान लेते हैं. हम मान लेते हैं our life cycle of ectocarpus is starting from this zygote. This zygote will develop into sporophyte. This sporophyte has two structures, plurilocular sporangia and unilocular sporangia. Plurilocular sporangia after mitosis will give rise to mitospore or mitozoospore or zoospore, और वे mitospore एक के बाद एक जनरेशन में sporophyte बनाते हुए जा रहे हैं. This unilocular sporangia will undergo meiosis and will produce myozoospore or zoospore. दोनों जगह zoospore लिखा है, लेकिन ये zoospore mitospore या diploid है और ये वाला zoospore आपका haploid है. और this zoospore will germinate and will give rise to gametophyte. और gametophyte में क्या होता है कि इसकी सेल्स आपको थोड़ी छोटी दिख रही होंगी compared to these cells. Okay, एक तो डिफरेंस है. In this gametophyte you will not find this unilocular sporangia, but plurilocular sporangia इसमें भी दिखाई देगी. And these plurilocular sporangia of gametophyte will produce male gamete and female gamete. Again, male gametophyte and female gametophyte, they are almost similar to each other, and it's not easy for anyone to differentiate on the basis of their morphology. तो उस बेस पे भी हमने इसको isomorphic type life cycle बोला है. Then when it is producing the male gamete and female gamete, this female gamete after 5 minutes it will get settled down and start releasing ectocarpin. एक हार्मोन रिलीज करेगा, इसको बोलते हैं ectocarpin. That ectocarpin will attack these male gametes, और ये मेल गैमेट्स कैसे आएंगे, उसके बाद एक सर्कुलर मोशन में आएंगे, घूमते घूमते बहुत तेजी से घूमेंगे, एंड जितना उसके पास आएंगे इनका मूवमेंट कम हो जाएगा. एंड ऐसा नहीं कि जहां तो हमने एक ही दिखाया है, बट एक्चुअल में क्या होगा, दे आर मैनी मेल गैमीट विल कम इन कॉन्टेक्ट ऑफ फीमेल गैमीट, और जो उनका लॉन्ग फ्लैजेला है, हेटेरोकोन्ट में 2 होते हैं ना, एक छोटा एक बड़ा, द लॉन्ग फ्लैजेला, with the help of this long flagella they will get attached themselves with this female gamete. Okay, and one of these like sperm and egg का जो fusion होता है, only one sperm will get fused with the egg cell. Same way only one of these male gamete will get fused or will be successful to fuse itself with the female gamete, and once they get fused it will become zygote. It will stop production of ectocarpin, और एक्टोकार्पिन का प्रोडक्शन जैसे ही बंद करेगा, all those flagella which were attached apart from that one which had already fertilized, एक जो फर्टिलाइज हो चुका है उसके अलावा बाकी सारे अपने आप को डिटैच कर लेंगे इस पूरे इससे, फीमेल से, एंड देन दिस डाइगोट विल जर्मिनेट इन नेक्स्ट फ्यू डेज। इस पूरे केस में हम क्या एक ही चीज स्किप करके आए थे कि सम टाइम इट इज पॉसिबल दैट दी गैमीट्स, यहाँ पे फीमेल दिखा है, मेल और फीमेल may directly germinate instead of fusion, and this development is known as parthenogenesis। ठीक है, चलो अगर कुछ और पूछना है तो पूछ सकते हैं मुझसे, किसी को डाउट है तो। So this is the classification as proposed by Lee with justification. Why it is under ectocarpus? Thallus consisting of filament or filament compacted together, as well as their reproduction is isogamous or anisogamous. These are the diagrams you have to make. Thanks to Minal ma'am, उन्होंने सारे अपने diagrams दे दिए। तो अगर आपको original diagrams या book से बनाने, it will take too much time. What you have to do, this is showing how it will look like when you are observing it in the low power. एक सिंपल सी ब्रांच दिखेंगे एंड बीच बीच में आपको स्पोरेंजिया ये दिख रहे हैं, प्लूरिलोक्युलर स्पोरेंजिया इस तरह से दिखाई देंगे। इफ यू विल enlarge it one portion, you will see that it is made up of single row of cells, एक के बाद एक cells जुड़ी हुई हैं, and plurilocular sporangia is also visible. It's a thallus morphology, it's a part of thallus. इसके बाद this is the enlarged vegetative cells. Only these things are visible to us. एक न्यूक्लियस हमें दिख रहा है कहीं कहीं पर और क्रोमेटोफोर्स हमें दिख रहे हैं. We are not able to see any other details. Then a unilocular sporangia and plurilocular sporangia. Simple diagrams बनाने हैं just to understand how and where they are located and how it will look like. इसके अलावा sometime it is also coming that why Ectocarpus is coming out an emerging model organism. Model organism का आपको पता है कि it's short life cycle, metabolic activity, as well as it is leading the sex hormone which can be easily detected in the culture system. इसका जो ectocarpin है, it is not restricted to one species. Like what do I mean to say? Okay, suppose there is one species from where you have collected the ectocarpin and tried to attract the male gamete of other species of Ectocarpus, so it will work over there as well. This is the best example to see the alternation of generation. Both are equally dominating, gametophytic as well as sporophytic, and they are alternating with each other. To understand the life cycle, alternation of generation, to understand the metabolic activity, to understand the chloroplast structure, to understand the haplodiploid life cycle, to understand the viral infection, to understand the host-parasitic interaction. There are so many things. Because of that, Ectocarpus is one of the easiest material to study. That's why it is coming out an emerging model organism. Like Chlorella की अपनी खास बात है, Spirulina की अपनी खास बात है, Chlamydomonas की अपनी खास बात है, but along with all those things, Ectocarpus has something very unique. It's not easy to find out and collect and isolate the pheromone, but it's very easy compared to any other algae or plant. It's very easy in case of Ectocarpus. Because you can also find it as an epiphyte or endophyte. So you can also see the relationship between the host parasite. Means there are certain virus which infect the Ectocarpus as well. So viral infection on Ectocarpus or Ectocarpus attachment on some other algae can also be easily studied with the help of this. 2-3 महीने के अंदर ही आप पूरी लाइफ साइकिल को एक के बाद एक स्टडी कर सकते हैं. It means if you will spend your 2 to 3 years, you will get lots of data, lots of information about that specific species of Ectocarpus. So that's why it is coming out an emerging model organism. If you want to know more about Ectocarpus, तो एक है डॉक्टर आलोक अरुण जो पोर्टोरिको में, एंड ही विल बी हैप्पी टू गिव यू मोर इंफॉर्मेशन अबाउट द एक्टोकार्पस। अगर कोई आजकल कभी भी आपको इंफॉर्मेशन चाहिए एक्टोकार्पस के बारे में तो, या फिरोमोन्स के बारे में, तो ही इज द बेस्ट पर्सन जिसको आप अप्रोच कर सकते हैं। There is one more slide I will not discuss. This is the additional information when and where gametophyte will develop, when and where sporophyte will develop. I am giving you this slide is showing what are those environmental factors which help in the development of a particular stage. कि ये अब स्पोरोफाइट डेवलप होएगा या गेमेटोफाइट डेवलप होएगा, यूनिलोकुलर स्पोरेंजिया डेवलप होएगी या प्लूरिलोकुलर स्पोरेंजिया डेवलप होएगी। तो in this by this paragraph you will try to understand that yes, there are temperature, salinity, and pH level is there, so that we can also control कि अभी हमारा plant sporophyte बनाए या gametophyte बनाए। But it's all these are additional information. Students, कोई डाउट, क्योंकि थोड़ी सी ज्यादा चीजें इसमें बतानी पड़ी हैं, बाकी समय तो क्या था आराम से हैप्लोटिक लाइफ साइकिल चल रही थी हमारी, और अलग बिल्कुल इट वाज ए डिफरेंट मेथड ऑफ सेक्सुअल रिप्रोडक्शन। लेकिन इस केस में क्या होता है, इन केस ऑफ एक्टोकार्पस बोथ द थिंग्स आर इंटेंगल्ड विद ईच अदर, एसेक्सुअल हो या सेक्सुअल हो, सब एक साथ ही लाइफ साइकिल में चल रहे हैं। तो उसको आपको समझना होगा कि सेक्सुअल का मतलब केवल वही है when gametes are getting fused. This is the part of sexual life cycle. All other things which are happening during the life cycle, they are mainly the part of asexual life cycle. क्योंकि आपका जो न्यूजिस्पोर बनना है ये जरूरी है सेक्सुअल लाइफ साइकिल से बनने के लिए, बट जरूरी नहीं कि इसके होने के बाद सेक्सुअल लाइफ साइकिल में जाए। दे कैन टेक ए पाथ ऑफ पार्थेनोजेनेसिस। तो ये समझ लीजिए कि इसका मेन पाथ तो सेक्सुअल लाइफ साइकिल है, लेकिन दे आर मेनी ऑल्टरनेट रूट। जैसे आपको सपोज कीजिए आपको कश्मीर से कन्याकुमारी जाना है, तो सीधा रास्ता तो कश्मीर से कन्याकुमारी है, लेकिन आप हो सकता है कि कश्मीर से दिल्ली आए, दिल्ली से कलकत्ता होकर फिर भोपाल आके फिर कन्याकुमारी जाए, या फिर जब आप सीधे भोपाल चले जाएं या फिर नागपुर चले जाएं, एंड देन यू आर टेकिंग ए लॉन्ग रूट from Mumbai or somewhere else, Bhubaneswar, like that. So the main route is quite simple. Sporophyte will give rise to myospores by the help of unilocular sporangia, which will germinate and give rise to gametophyte. Gametophyte will give rise to gamete, get fused, make zygote, will develop into sporophyte. Simple सा रूट ये, लेकिन स्पोरोफाइट से भी स्पोरोफाइट बन सकता है क्योंकि ये माइटोस्पोर बनाते हैं। इस गैमेटोफाइट्स के गैमीट से भी गैमेटोफाइट बन सकता है क्योंकि दे कैन टेक द पाथ ऑफ पार्थेनोजेनेसिस। ओके, तो ये अलग अलग रूट्स हमारे अवेलेबल हैं एक्टोकार्पस में। बच्चों बताओ, कुछ पूछना है तो? अगर आपको किसी को भी लगता है कि जो ड्राइंग से दे आर नॉट द ट्रू रिप्रेजेंटेशन ऑफ व्हाट वी हैव ऑब्जर्व्ड इन द लाइव मटेरियल और द फिक्स्ड मटेरियल, then you can also make it after the discussion on your own way. Otherwise ये बहुत सिंपल डायग्राम से आप इनको डायरेक्टली बना लीजिएगा। This is the enlarged view and these are the unilocular and plurilocular sporangia. Thank you।